Hey guys, welcome back. In this session, we are discussing last 20-year immunology questions which came in GATE Biotechnology paper. So let's get started. Question number 31 says the first humanized monoclonal antibody approved for the treatment of breast cancer is Herceptin in combination with the chemotherapy drug Taxol is approved for the first line treatment of human epidermal growth factor receptor 2 positive, that is HER2 positive. So answer D, Herceptin is the right answer. Question number 32 says the role of an adjuvant is to adjuvants are compounds that enhance the specific immune response against co-inoculated antigens. Adjuvant can be used for various purposes. First, to enhance the immunogenicity of highly purified or recombinant antigen. Second, to reduce the amount of antigen or the number of immunizations needed for protective immunity. Third, to improve the efficacy of vaccine in newborns, the elderly or immunocompromised persons. Fourth, as antigen delivery system for the uptake of antigens by the mucosa, etc. So option A, prolong the persistence of antigen is the right answer. Question number 33. Endogenous antigens are presented onto the cell surface along with. So endogenous antigens are internally generated molecules that become presented on the cell surface in the complex with MSC class 1. So option B, MSC class 1 is the right answer. Question number 34 says, which one of the following is an ABC transporter? So multi-drug resistant protein MRP is a broad specificity primary active transporter of organic anions conjugates that confers a multi-drug resistance phenotype when transfected into drug sensitive cell. So option A, multi-drug resistant protein is the right answer. Question number 35 says match the antibiotic in group 1 with the targets in group 2. So sulfonamide, the target of sulfonamides and the basis for their selectivity is the enzyme DHPS in the folic acid pathway. Quinolones, they have antimicrobial activity because of their action against the topoisomerase enzyme. Erythromycins, they are used to prevent peptide chain elongation, and cephalosporin, they inhibit peptidoglycan synthesis. So option D, P3Q4R2S1 is the right option. Question number 36 says prior exposure of plants to pathogen attack. This phenomena is known as systemic acquired resistance is a mechanism of induced defense that confirms long-lasting protection against a broad spectrum of microorganisms. So option A, systemic acquired resistance is the right answer. Question number 37 says reaction between antibodies and antigens that are detected by precipitate formation in an agar gel are referred as. So immunodiffusion in gel are classified as single diffusion and double diffusion. Both antigens and antibody are allowed to diffuse into the gel. This assay frequently used for comparing different antigen preparation. So option B, immunodiffusion assay is the right answer. Question number 38 says cholera toxin increases CAMP levels by. So cholera toxin consists of an A subunit coupled to a B subunit. A subunit enters the cytosol where it activates the GS protein through an ADP ribosylation reaction that acts to lock the GS protein in its GTP bound form, thereby continually stimulating adenylate cyclase to produce CAMP. So option B, modifying GS protein is the right answer. Question number 39 says match the item in group 1 with group 2. So tyrosine kinase is used to receive insulin hormone signals and regulates the level of insulin in the body. Cyclic GMP is part of 7 helical transmembrane domain protein comprising of G-coupled receptor whose receptor protein is guanylyl cyclase. GTPase activating protein or GAP protein is used for the inactivation of G protein and stop the signaling. Nuclear receptor receives thyroid hormone as a signal for signal transduction. Hence option D, that is P2Q4R1 S3 is the right answer. Question number 14 says match the immunoglobulin class in group 1 with its property in group 2. So IgG, it is the antibody present in the highest concentration in the serum. IgA, it is the major antibody in external secretions. IgE protects against parasites, and IgM, it is the antibody that appears first in serum after exposure to an antigen. So option A, that is P4, Q1, R2, S3 is the right option.